Welcome back. In the last class, we talked about carbohydrates and lipids. Today, we're going to finish off our discussion of the macromolecules by discussing proteins and finally nucleic acids. By the end, you should be able to describe the structure and function of all four of the macromolecules. So proteins are a versatile macromolecule. They serve as a building block for a lot of the structures within our bodies. They form the basis of keratin, which makes up hair and nails. They form as storage in terms of myoglobin. Myoglobin is found in muscle and helps to store energy for use over a long period of time, sustained use. Proteins are important for regulatory issues such as hormones like insulin. They make up enzymes, which help us to speed up or conduct reactions at a lower temperature. And they're important for transport, such as hemoglobin being able to transport oxygen throughout the bloodstream. There are 20 different amino acids that are going to make up the proteins, and they are strung together the same way that letters are strung together to make words, to make up different proteins. The order in which the amino acids are bonded are going to result in different proteins. So this is what each amino acid is going to look like. It's going to have a central carbon, and if you remember, carbon has 4 potential bond sites. One of those bond sites is going to have a hydroxyl. This is, a carboxyl, which is a carbon double bonded to an oxygen and bonded to an additional oxygen. Sometimes it will be the carbon bonded to an oxygen, bonded to another oxygen, which is then bonded to a hydrogen. So sometimes it will be COO with a double bond. Sometimes it'll be CO and then OH. And this is your carboxyl group. There's going to also be an amino group. Notice for the first time we have nitrogen. The carbohydrates and lipids did not have nitrogen, but they're essential in amino acids. They form the amino group, which is going to be a nitrogen bonded with 2 and sometimes 3 hydrogens. The third bond site for the carbon is going to be a lone hydrogen atom. And then the fourth one is going to be an R group, or a side chain. And this is going to be different in each amino acid. And it's whatever is found in the location of this R group that's going to result in the 20 different amino acids. So you always have an amino group with a nitrogen and 2 or 3 hydrogens. You're going to have carbon bonded to a lone hydrogen, and you're going to have the carbon bonded to a carboxyl group with a carbon double bonded to an oxygen, or carbon bonded to an oxygen, bonded to a hydrogen, and then bonded to a second oxygen. Proteins are an essential part of our diet because we are not capable of producing all 20 amino acids, and yet we need proteins and the availability of all 20 amino acids for growth, repair, and replacement in order for our bodies to grow, to heal, and to replace lost cells. So it's important that we have a complete protein. A complete protein has all 20 amino acids and is very common in animal products. Incomplete proteins include beans and various other substances like nuts, and they may have a subset, either a large subset or a small subset of these amino acids, but they do not have all of them. So it's important, especially for vegetarians, to consume complementary proteins, to mix 2 different foods that each lack some amino acids to make sure that together they get all of them. So in the South, we tend to eat rice with black-eyed peas or corn and beans, and very commonly in Indian foods, they'll have rice with lentils. And each one of these is a format of getting complementary proteins that are going to make available to your body all 20 of the essential amino acids. So there are 2 questions on the next 2 slides. Pause the video and think about them for a few minutes, and then come back and start the video. Okay, so hopefully you've answered this and realized that the reason it's not sufficient to have just the amount of protein is that it might have a large amount of protein but not be complete. It might lack some of the essential amino acids, and the consumer should know this, especially if they're vegetarian. And for the second one, and hopefully you realize that both a hamburger or corn plus a legume, a bean, which are complementary proteins, would give you all of the essential amino acids that you need in your diet. So proteins, just like any other molecule, is going to be strongly influenced by its shape. Now for the first time with the proteins, we're going to be discussing a three-dimensional shape. There are 4 different components to the structure of a protein. And just like you saw with dehydration synthesis with the carbohydrates and the lipids, you're going to see that dehydration synthesis is going to be responsible for holding various amino acids together to form the protein. And so as we look at the 4 different structures, the primary structure is just the order of the amino acids, essentially how you spell out that protein, which amino acids are going to go in which place. Then the secondary structure is how this string of amino acids, how this protein is shaped. Is it going to form a helix, which is called an alpha helix? Is it going to have folds and kinks, and that's called a pleated sheet? These are 2 of the possible structures. So primary structure is the order of the amino acids. Secondary structure is how that link of proteins are shaped. The tertiary structure is how it's folded over on itself and which amino acids are going to bond with which through a series of hydrogen bonds. And then the quaternary structure, the 4th level structure, is how the different amino acids are going to bond with one another. So 4 different elements which are going to form a 3-dimensional shape and make each protein unique. And as we can see, we can denature a protein. We can stop it from functioning the way that it had by changing the shape, and there are a number of ways of doing this. 2 of them are by changing the pH or by changing the temperature. So if we take a protein like a hunk of ground beef and we begin to cook it, it's going to denature, it's going to change the shape, and you'll notice that it will change from that sort of red-pink color to a brown color. That's indicating visually that it has changed. The smell is also going to change, and you can tell that it has been completely denatured. Another way of doing this is with pH, and there's a common South American dish called ceviche where they will take fish, and rather than cooking it by using heat, they will cook it by using acid, and they'll put it in a bath of essentially lemon juice mixed with onions and cilantro and a few other things to make it taste good, and leave it in a cool refrigerated place overnight, and the acids itself will denature, will break down the proteins, and it will be cooked, not cooked with temperature, but cooked with acid. So why is it that egg whites are going to change their texture if you beat them? If, say, you're trying to make frosting. Okay, and it's because when you beat it, you're going to alter the shape. It's going to essentially stop becoming that particular protein and become a different protein. It's going to change its function. And which is most important? Okay, and hopefully you realized it was not the both. This is the first time I've done one that didn't have both as the answer. But the shape of a protein is on what the function is dependent. And as I said, proteins are important for enzymes. They're going to either begin a reaction, or they're going to speed it up, make it happen faster, or they're going to let it happen at a lower temperature than it might be able to happen otherwise. Lactase is one such enzyme. It's going to take lactose, which is a carbohydrate that is found in milk. It is comprised of, it's a disaccharide. It has two monosaccharides, galactose and glucose, making it up. And the enzyme lactase, ACE is always going to let you know that you have an enzyme. Lactase is going to allow the bond between the glucose and the galactose to be broken. If you don't have lactase, then you can't break that up, and it's difficult or impossible to digest milk. And the place in the enzyme where the specific molecule is going to land is called the active site. And the active site is generally unique. It can only function with one particular molecule, occasionally with a very small range of molecules. And of course, the absence of lactase or the inability to produce it is why some people, especially as they get older, tend to lose the ability to digest milk. They can sometimes supplement by taking a lactase pill, and that provides the body with the necessary enzyme to break the milk or other dairy products up, but sometimes even that doesn't work. And of course, if you don't break it up, which the milk is going to pass through your stomach and pass through your small intestines and get into your large intestine without having ever been digested. In the large intestine are a series of bacteria, and the bacteria will begin to break it up. But when they do, they release a series of gases, and it causes both bloating and discomfort, as well as, production of gases that will then be released from your body and are also unpleasant. Okay, so why is it that adults lose this ability, or some adults lose the ability to process milk, and why is it that some children are born with the inability to process milk? And that's because when the body gets the instructions for how to build that enzyme, that particular protein, essentially it comes programmed with a spelling error. And instead of being placed in the proper order, the amino acids are placed improperly, spell the wrong protein, and form an enzyme that winds up not being effective. And the same as with lactose intolerance, fetal ketonuria is a far more serious inability to break down bodily wastes.